In this next video, we're going to be talking about the current status of biological diversity. If we want to conserve biological diversity, we need to have a starting point. We need to know what we're working with. What should we be aiming to preserve? What is the current biological diversity on Earth that requires conservation? Seems like a straightforward question. It's more tricky, or it's more complicated than you might expect. So, you might have heard talk about how we are currently in the midst of the 6th major extinction event. It's a term or a phrase that gets thrown around quite a bit, and one that is actually subject to some debate. For your interest, I've posted 2 different articles on arguing opposite sides of this question. Are we in the midst of the 6th major extinction event? One argues yes, the other argues no. Regardless of whether you classify our current extinction rates as a major extinction event, again, recall the lecture way back another lifetime ago on extinction. We talked about the 5 major extinction events in the geological record. Whether or not you characterize current extinction as comparable to those other big 5 extinction events is a matter of some debate. But what is not up for the debate is the knowledge that we are resulting in the extinction of biodiversity on Earth at alarming rates, and something that needs to be reversed to maintain environmental or ecosystem health. The first scientist to really sound the alarm bell on high rates of extinction, or the first one to get any notice for doing so anyway, was E.O. Wilson. You might recognize Wilson from the equilibrium theory of island biogeography, Wilson and MacArthur. But he was also spent a lot of his effort calculating biodiversity loss. And he calculated that the biodiversity loss of species from the tropical forest area alone could be as high as 6,000 species per year, which would, in his view, put us in the 6th major extinction events. And recent extinction rates may be as many as 100 to 1,000 times greater than those before the Anthropocene, wherever you set the Anthropocene boundary. So, if we are losing species at an alarming rate, how many species currently exist? How many species do we have to lose, in other words? So we currently have 1.8 or approximately 1.8 million species that have been described on Earth. But estimates actually range between 3 and as many as 500 million species. So what this tells us is that only a very small proportion of the total number of species on Earth have been adequately described. So there are many, many more species out there that we don't know about. And if we don't know about them, then we can't do much to conserve them. So 2 examples of relatively newly described species. It's quite common to discover new species of insects. The example that I have here is a new moth that was discovered from museum specimens. But we're still, surprisingly enough, we are still discovering new species of mammals. So mammals have received much more attention in conservation, biology, biogeography, zoology studies. You might think that we've described all the mammal species currently living on Earth, but we actually haven't. One example of a new mammal species that was recently discovered is a new species of gibbon that was discovered in China. And this is actually a population that had been studied for some time until new research really revealed that it was actually a different species. It was thought to be the same species as other populations of gibbons being studied, and it wasn't until we had some capacity to look at the genomic structure of these populations that scientists realized that this was actually, in fact, its own species. So that is what we call a cryptic species. The gibbon is an example of a cryptic species. So cryptic species are individuals that appear morphologically identical to another species, but are actually genetically quite distinct, and they may even have reproductive barriers that are unknown or not apparent when you look at morphology alone. So the new species of gibbon on the previous slide is one example of a cryptic species. The other example that I have here is a species of water flea. So this tiny little globular invertebrate here that I have a picture of is from a group known as Chidorus sphericus. And Chidorus sphericus used to be thought of as the true cosmopolitan species. It was found It's found on every continent. It lives in lakes on virtually every continent. But once we started to do some genomic mapping, to start looking at the genetics of Chaedoros sphericus, we actually discovered that Chaedoros sphericus is not a species, but it's a species complex. So it's a group of species that have their own individual species representation on the different continents. So it looks like Chaedoros sphericus in North America is a different species from what looks like Chaedoros sphericus in Europe, in Australia, and South America. So a cryptic species. You can't tell just by comparing the morphology, but when you look at the genetic structure, it's genetically distinct. So that brings us to a challenge that we have in conserving biodiversity called the Linnaean shortfall. The idea that most species that currently exist on Earth are unknown to science. And the implication of this is that we are vastly underestimating the global decline in biodiversity due to human activities. Another way to refer to this is centennial extinctions. So the idea that undiscovered species go extinct before they're even known to science. So we're losing species that we don't even know about. Another type of challenge or shortfall for conserving biodiversity, and one that biogeography is uniquely suited to address, is what we call the Wallacean shortfall, named after Alfred Wallace. And that's the idea that our knowledge of the geographic distribution of most species is incomplete, and particularly in highly diverse tropical ecosystems. So quite simply, our sampling effort is not where it needs to be in order to identify what species live where. So I study lakes, I'm a limnologist, and we have a saying in limnology that the earliest limnology studies was the study of lakes that were a stone's throw from a university campus. The idea that for logistical reasons, we tend to study regions that are easy to get to, are easy to access. And the more remote regions are going to be understudied. So we know a lot more about the northern hemisphere than we do about the southern hemisphere in terms of biogeography. So when we're talking about conserving species, we want to conserve a species range or a species distribution, but we don't actually know what those species distributions are. So we might think that a species is endemic to a very small area, but maybe that's just a reflection of where we've been sampling. We found it in that area because that's where our sampling effort has been placed. And the areas adjacent to that where we haven't been sampling may contain representatives of this species as well. We just don't know about it. So again, just in summary, the Wallacean shortfall is a shortfall in our sampling effort. It's the idea that large areas of the Earth have not been explored for their species diversity. Their biogeography has not been adequately characterized. We have incomplete data for characterizing species ranges, which presents challenges for conserving those species. So this ties into species distribution modeling. So species distribution modeling, you've seen those maps of species ranges, the areas that we find as given species on the globe. Well, those are based on a species distribution model. So to develop a species distribution model, you would go out and you would sample given geographic regions, and you would look and identify where you find that species and where you don't. So you would have what we call presence-absence data, where a species is found and where it's not found. And then you would map that. And then onto that map, you would overlay other variables that might explain that distribution. So for example, you would put temperature data over top that, precipitation data, habitat data, and you would use that to start to get an understanding of why species are found in some areas and not others. And these species distribution models are incredibly important for modeling how climate change is going to impact species. So if you identify a certain bioclimatic envelope, that is a climate that is favorable to a given species, we know that the climate is going to change. We can make projections about how the climate is going to change. So we can identify where suitable climate for that species is going to be found in the future. And we can map, so you can see, is there a capacity for that species to track that habitat with climate change. But if we have incomplete data, then our models on how species are going to change with climate or with other human modification of the landscape is going to be incomplete. It's going to be based on faulty data. So we have another saying that is basically garbage in, garbage out. The idea that if we're making a wide number of assumptions and we have uncertainty in our data, so if we've only looked at a small geographic area, scaling that out to a wider geographic area and developing these species range maps are not going to be very accurate if we don't have an intense enough sampling effort within that area. So basically what that means is when we're saying that a species is absent from a given area, is it actually absent or did we just not sample that area well enough? Absence of evidence is not evidence of absence, is another saying. So garbage in, garbage out. Evidence of absence is not absence of evidence is not evidence of absence. Those are two sayings to keep in your mind. So if we're evaluating these species range maps, and you've seen a lot of them in this course already, it's important to ask about the quality of the data underlying that. So when we look at a species range map of lake trout in North America, like I have here, we can have a high degree of confidence that this map is accurate. North America has been extensively studied, and lake trout are a very popular recreational sporting fish. People like to go out and catch lake trout, so there'll be lots of data on where you find them and where you don't. But if we're going to map a species that people don't care about as much in a region of the world that isn't as well studied, the range map that we develop might not have the same kind of rigor or the same confidence associated with it. So, in summary, if we want to conserve native species, we need to conserve their habitats. We need to conserve their distributions. And since biogeography is the study of what species live where and why, conservation biogeography is uniquely suited to conserve species distributions.